This is chapter 10, section 2, and this deals with cell division. So we're going to go through IP-MAT-C. And we want to start with, this is a human karyotyping, and what we do is we use what's called electrophoresis to put chromosomes through. And as we put chromosomes through, these would be the largest homologous pair of chromosomes. So we call those chromosome pair number 1 or homologous pair number 1. Notice that as we move through this human karyotyping, the sets of chromosomes or the homologous pair of chromosomes get smaller. And right down here, this is our X and Y chromosome. This is a male human karyotyping because we have an X chromosome and then a smaller chromosome here, which is the Y. Humans have 23 pair. The first 1 through 22 are called autosomes, and the last pair are called sex chromosomes. So this, again, is a human karyotyping, and we have 46 total chromosomes. We get 1 of each of these from our mom and 1 of each of these from our biological dad. And what these are is DNA tightly wrapped up around these proteins called histones. So that protein in DNA will form a chromatid, which is right here. Chromatid, one side. This is the sister chromatid to that. And these are held together by what's called the centromere. So if you look right here, this little X shape right where the cross meets or the chromatids meet, that's called the centromere. Our spindle fibers will hook up to that, we'll notice here pretty soon. At the end, you can, what you'll see are little proteins that make what's called a telomere, and they look to me like little stocking caps that you see at the tip of each chromosome, so telomere. And every single organism has a specific set of chromosomes. And just because we have 46 doesn't mean we are the all-dominant species. There are species such as a chicken, which has 78, that actually have more chromosomes. Some of the plant kingdom have the highest. There are ferns with over 1,000 chromosomes. And plants tend to have more mutations than we do, and it's one way that plants change throughout. We can only see chromosomes, so these right here, besides when we do electrophoresis or put them in a human karyotyping or some kind of karyotyping for another creature, other than that, we can only see them during prophase, metaphase, anaphase, and telophase, so only during cell division. And then each chromatid is identical to the sister chromatid. So this chromatid is identical to this chromatid. And again, each identical half is called sister chromatid. So sister chromatid, sister chromatid. Each one of these chromosomes has 2 sister chromatids. This is a cell cycle. Notice that interphase is the longest cycle. Most of the cells that we have viewed underneath microscopes are interphase. And on top of that, most cells stop at the G1 phase and maintain a certain cell size. If there's a need, we will enter into the S phase where we copy DNA, and then we'll move in to prepare for mitosis or the M phase. Notice the M phase is mitosis, prophase, metaphase, anaphase, telophase, plus cytokinesis. You may also see that called cell division. So again, in interphase there are 3 phases or subphases. The G1 for growth, the S for DNA replication, and then we will call the G2 preparing for mitosis. So the 2 stages. The M phase or cell division, again, remember this is prophase plus cytokinesis. Mitosis is the division of the nucleus into 2 nuclei. So we went from 1 cell here, and now we have 2 nuclei. And remember, we'll have the exact same genetic information in each one of these, whether it's an animal cell or a plant cell. And then cytokinesis is what you see here. You'll see that there's a cleavage furrow forming here, and these 2 animal cells will eventually split in cytokinesis. A plant cell will actually do what's called build a cell plate or build another cell wall when it does cytokinesis. So again, M phase is mitosis plus cytokinesis. So the cell cycle, or the series of events that a cell goes through, they're labeled right here. Again, the G1, the S, and the G2 are all under interphase. Most cells stop somewhere at this point. The M phase is prophase, metaphase, anaphase, telophase, plus cytokinesis. And as the cell grows, it will if needed, depending on where the cell is, prepare for division and starting with the S phase and move into the G2 phase. You may also see these called the gap phase. And eukaryotic cells, and this cell cycle is specifically for eukaryotic cells. Prokaryotic cells end up doing a little bit different process. So the cells divide to form two cells when they're needed, and skin tend to be one of the types of cells that prepare for this more readily than other cells. So what is this? This is interphase. Again, interphase has three subphases. Most of these cells that we're looking at here, like these guys in interphase and these guys right here in interphase, they are in and will always be in the G1 phase. Some of them, which we can't really see because it's an interphase, will begin the S phase, and we're going to make copies of those sister chromatids, make chromosomes, basically copying DNA. And then the G2 phase, we're going to prepare for the next step, which is prophase. Do remember that interphase is not part of mitosis. Again, for review, we just went through interphase, growth, DNA replication in the S, growth is D1, S, DNA replication, G2 is preparing for mitosis. Now we're going to get a little bit more into these. So once we start cell division, we can see chromosomes in these four phases and kind of a little bit in cytokinesis. The four phases of mitosis are prophase, metaphase, anaphase, and telophase, and then cytokinesis is added to that. Prophase. Again, we're talking about cell division. Prophase will be the first step in the M phase. It's mitosis plus cytokinesis, and the cell will divide into two daughter cells with the same genetic information. Prophase is the longest phase of mitosis, and what happens is we're starting to form these spindle fibers, chromosomes are starting to appear. If it's an animal cell, these centrioles will migrate to opposite ends. So again, you'll see chromosomes, nucleolus. Notice there's no darkened spot of chromatin anywhere. It's gone. The nuclear envelope is broken apart. Again, as I said, the centrioles are moving to opposite ends of the pole, and these right here are spindle fibers. Those are those microtubules or microfilaments. And then those centrioles will move to the other end of the poles, and these spindle fibers will attach themselves to the centromeres of the chromosomes. Metaphase, which is the shortest phase? It's right here. We line up chromosomes on what's called the metaphase plate. Plants, animals, any eukaryotic cell does this, where we line up on the metaphase plate. This is done by those centromeres attaching to those fibers, and then you can see that they're kind of taut, if you will, and right before you can actually see where they pull, and then all of a sudden we'll move into the next phase, which is anaphase. And anaphase, those centromeres will pull apart the sister chromatids. Okay, so each one of these is a sister chromatid, and what I was looking for when we were doing the phases were these sister chromatids pointed toward each other. You can see that they're pointed toward each other, pulling apart. When the movement stops in this, when we're done pulling, anaphase is over. The next, telophase. So this is the final phase of mitosis. What starts to happen is the nuclear envelope will start to reappear. The chromosomes will, or excuse me, this should say chromatids start to uncoil. The nuclear envelope starts to reappear, so we're going to start making another nucleus. And those spindle fibers are starting to break apart to provide structure and strength for a cell. And then we'll start seeing a nucleolus start to appear. So again, that's telophase. And then finally, cytokinesis, the last step of the M phase, or cell division. In plants, again, you'll get what's called a cleavage furrow forming. It's where that cell membrane is pinching off. Cytokinesis means pinching off or separation of the cytoplasm. So notice we got a nucleus, or nuclei, excuse me, formed here. We're basically worried about separating the cell now. Plants will form a cell plate, so it's like little construction workers are bringing in a whole bunch of material to make another cell wall. So again, cleavage furrow in animal and cell plate in plant cells. And again, this is the last part of the M phase, but cytokinesis is not considered part of mitosis. So in review, there are two major, if you will, phases of the cell cycle. You got interphase, which will have the growth phase or the G1 phase. It may also move into the S phase, which is where we copy DNA, and the G2 phase is preparing for the M phase or mitosis. Mitosis also, we can't forget about this guy, cytokinesis. These two together equals the M phase or the cell division phase. Mitosis by itself is prophase, metaphase, anaphase, and telophase, and then the last step is cytokinesis. I hope that helps, and thanks for listening.